April 25th, 1953. On this day, a short scientific paper published in the journal Nature revealed the structure of DNA, providing for the first time a coherent explanation of how hereditary information is stored, copied, and transmitted in living organisms. This moment did not mark the discovery of DNA itself, which had been known since the 19th century. Nor did it instantly answer every question in biology, but it fundamentally changed the way scientists understood life, transforming a poorly understood molecule into the central blueprint of all living systems. To fully grasp the importance of this breakthrough, it is necessary to look back at the scientific landscape that existed before it. For centuries, inheritance had been one of the most obvious yet least understood features of life. People could observe that children resemble their parents, that certain traits reappear across generations, and that patterns of heredity exist within families. Yet no one could identify the physical mechanism responsible for these observations. Even after the rediscovery of Gregor Mendel's work in the early 20th century, which established the basic laws of inheritance, the question of what carried this information at the molecular level remained unresolved. By the 1940s, experiments had begun to suggest that DNA, rather than proteins, was the molecule responsible for genetic inheritance. But this idea was still not universally accepted. Proteins, with their structural complexity and variability, appeared to be far better suited to encoding biological information, while DNA seemed too simple, consisting of only a limited set of repeating components. As a result, many scientists remained skeptical, and the role of DNA continued to be debated. What was clear, however, was that understanding the structure of DNA would be essential, because in biology, structure is closely linked to function, and without knowing the form of the molecule, it was impossible to fully understand what it did. Among the most important experimental tools used to investigate molecular structure was X-ray diffraction, a technique that allowed scientists to infer the arrangement of atoms by analyzing how X-rays scatter when they pass through a crystalline or semi-ordered sample. At King's College London, Rosalind applied this method with exceptional rigor, producing highly detailed diffraction images of DNA fibers. Her work required careful preparation of samples, precise control of experimental conditions, and meticulous interpretation of the resulting patterns. One of her most significant contributions was an image later known as Photo 51, which displayed a clear X-shaped diffraction pattern that indicated a helical structure. Although the image itself appeared abstract, it contained precise quantitative information about the geometry of DNA, including the spacing between repeating units and the overall symmetry of the molecule. Franklin's data provided some of the strongest experimental evidence available at the time, even though its full implications were not immediately recognized in a broader theoretical context. At the same time, at the University of Cambridge, James Watson and Francis Crick were pursuing a complementary approach based on model building and theoretical reasoning. Rather than relying solely on experimental measurements, they sought to construct a three-dimensional model of DNA that would be consistent with known chemical principles and available data. By integrating information from multiple sources, including Franklin's diffraction results and established knowledge about nucleotide chemistry, they developed a model in which DNA consists of two anti-parallel strands that wind around each other to form a double helix. In this structure, the sugar phosphate backbones are positioned on the exterior, while the paired nitrogenous bases form the interior rungs of the helix, held together by specific patterns of hydrogen bonding. The importance of this model lay not only in its structural accuracy, but also in its ability to explain fundamental biological processes. The pairing of bases, adenine with thymine and cytosine with guanine, revealed a system of complementary matching that provided a direct mechanism for encoding information. The sequence of these bases functions as a linear code, a molecular language through which genetic instructions can be stored and interpreted. Equally significant was the implication for replication. Because each base pairs with a specific partner, the two strands of DNA are complementary, meaning that each strand contains the information necessary to reconstruct the other. When the strands separate, each can serve as a template for the formation of a new complementary strand, allowing DNA to be copied with remarkable fidelity. Although this mechanism would later be confirmed experimentally, the double helix model offered an immediate and compelling explanation for how hereditary information could be both stable and reproducible. The publication of this model in 1953 marked a turning point in the history of science, as it provided a unifying framework that connected previously separate areas of research. It established the foundation for molecular genetics, enabling scientists to identify genes, to understand patterns of inheritance with unprecedented clarity, and to investigate the molecular basis of diseases. Over the following decades, this knowledge led to the development of DNA sequencing technologies, genetic engineering techniques, and a wide range of medical and biotechnological applications, including diagnostic tools, targeted therapies, and forensic methods for identifying individuals based on their genetic profiles. Beyond its immediate scientific impact, the discovery of DNA's structure also had profound philosophical implications, as it revealed that life is fundamentally based on information encoded at the molecular level. Every organism, from the simplest bacteria to complex multicellular beings, relies on the same basic chemical system for storing and transmitting genetic instructions. This realization demonstrated the deep unity of life on Earth, showing that all living organisms share a common biochemical foundation and are connected through a continuous chain of genetic inheritance extending back billions of years. Perhaps the most remarkable aspect of this discovery is that the system it revealed had always existed, operating within every cell of every organism long before humans became aware of it. DNA had been guiding the development, function, and reproduction of life throughout evolutionary history, silently encoding and transmitting information across countless generations. The events of April 25, 1953 did not create this system, but they made it visible, transforming an invisible molecular process into something that could be studied, understood, and ultimately manipulated. In this sense, the revelation of DNA's structure represents more than a scientific breakthrough. It marks a fundamental shift in human understanding, demonstrating that life is not only a physical phenomenon, but also an informational one. It showed that the complexity of living organisms arises from the organization of simple molecular components into a coherent system capable of storing, copying, and interpreting information with extraordinary precision. April 25, 1953. Therefore stands as the moment when humanity first gained the ability to read the fundamental code of life, converting one of the oldest and most profound mysteries in science into a system that could be analyzed with clarity and rigor. It revealed that life is structured rather than random, ordered rather than chaotic, and ultimately understandable through the language of chemistry and information. At its core, this discovery showed that every human being is defined not only by physical matter, but by a vast and dynamic sequence of information encoded within DNA, a sequence shaped by evolution, preserved through inheritance, and continuously expressed within the cells of the body. This code connects each individual to their ancestors, to other living organisms, and to the broader history of life on Earth, forming a continuous thread that extends across time. And it is this realization, emerging from a single publication in 1953, that forever changed the way humanity understands life. revealing that within every cell lies not just chemistry, but information, a living, self-replicating code that defines what it means to be alive.